Hello students, welcome you all to a new video of Sunanda's tutorial. In this video we will discuss about the plant growth hormones, plant growth regulators and photoperiodism as well as phototropism. So in this series it is the lecture video 1, and today in this video we will discuss about the first hormone, that is auxin, which is the first discovered plant hormone. So what is plant growth regulators? Regulator means which regulate the growth of the plant. Okay, and these are small organic compounds of diverse chemical composition, because there are many plant growth regulators: oxygen, cytokinin, gibberellin, ethylene. Okay, so these are plant growth regulators, abscisic acid, which influence the physiological activities of the plant at very low concentration. Okay, plant growth regulators are also called plant hormones or simply phytohormones or plant growth substances. On the basis of their function inside the living plant body, the plant growth regulators or PGRs or phytohormones are classified into two broad groups. What are those two groups? One is plant growth promoter, which shows positive activities, and plant growth inhibitors, which shows negative effect on the plant. Okay. So plant growth promoters, they are involved in growth promoting activities like cell division, cell enlargement, trophic growth hormones, trophic growth movements of the plant like photoperiodism, then flowering, then fruiting, fruit formation, seed formation. So what are the examples of plant growth promoters which affect the plant in positive sense? Those are auxin, gibberellin, cytokinin. Plant growth inhibitors, which involve in plant growth inhibiting activities like dormancy, abscission, like that. Example, abscisic acid and ethylene. Okay. So these are two groups of plant growth regulators. So at first we will discuss one of the plant growth promoter, which is auxin. Auxin was first phytohormone to be discovered. Charles Darwin, along with his son Francis Darwin, first observed that the coleoptile tip of a plant canary grass responded to the unilateral light. What is unilateral light? So this plant responds to unilateral light, that means light from one direction. Okay, so unilateral light, this canary grass respond to the unilateral light by bending towards the light source. Later it was found that tip of coleoptile is source of auxin that caused that bending of coleoptile. Next, F.W. Wendt in 1928 isolated auxin from the tip of coleoptile of Avena sativa, oats seedling. Okay, the most common naturally occurring auxin is indole acetic acid, IAA, that resembles the amino acid tryptophan from which it is synthesized. Okay, so IAA is synthesized from tryptophan, which is an amino acid. Next, IAA synthesized transport precipitally, that means from apex to the base through polar transport by parenchymatous cell and non-polar transport by phloem. Okay. The synthetic auxins are naphthalene acetic acid, NAA, IBA, indole butyric acid, 2,4-dichlorophenoxyacetic acid, 2,4-D, 2,4,5-T, 2,4,5-trichlorophenoxyacetic acid. So these are synthetic auxins. So what are the physiological effects of auxin? What they do inside the plant body? They stimulate cell elongation at first. They control cell division in vascular cambium. What is vascular cambium? From where the secondary growth starts. This auxin also stimulates xylem differentiation. Okay. Then auxin prevent premature fall or abscission of leaves, fruits and flowers. Okay. In higher plants, the apical bud suppress the growth of lateral or axillary bud. So this phenomenon of the suppression of lateral or axillary buds by apical bud is called as apical dominance. That means dominance of the apical part of the plant. If we remove that apical part or decapitation, it causes the sprouting of lateral buds. Therefore, decapitation widely applied in tea plant and hedge making. Okay. This auxin promotes the growth of stem and coleoptile but inhibit the growth of roots. It causes the bending of coleoptile towards slide. So it promotes phototropism but control geotropism. Okay. It promotes the production of female flowers. Together with ethylene, it shows positive effect towards the production of more female flowers in plant. Next, uses or applications of auxin in practical or day-to-day life. So at first, it is used to initiate rooting in stem cutting during vegetative propagation. Exogenous application, if we apply auxin exogenously, naphthalene acetic acid 2,4-D, that means 2,4-dichlorophenoxyacetic acid induce flowering in pineapple and lychee. Then 2,4-D, 2,4-dichlorophenoxyacetic acid and 2,4,5-trichlorophenoxyacetic acid used as herbicide to kill the dicot weeds but does not affect the monocot plant. Then indole acetic acid and indole butyric acid, these are used to produce seedless parthenocarpic fruits as in tomatoes. Banana is naturally parthenocarpic due to higher internal production of auxin in carpel stage. So these are practical application of auxin phytohormone. So with this, I am concluding my lecture video here on the phytohormone auxin. It is the first or part 1 video of phytohormones or plant growth hormones or plant growth regulators. In my next videos, I will discuss other plant growth regulators like gibberellin, cytokinin, ethylene, abscisic acid. So please stay tuned. If you liked it, please hit the like button, share with your other friends and please subscribe to my channel Sunanda's tutorial. Thank you.